120,000 proteins from 25,000 genes. That's based on how the cell is perceiving its environment. And I'm going to show you several experiments that's going to explain this. But you can start to see already, it's not just the DNA. There's something more to this. There's something that's driving the choices of which genes to use and which proteins are made. So this is a profound statement. It's from an article called Metaphors in the Role of Genes in Development. It came out in 1990 from Dr. Niehaus. When a gene product is needed, a signal from its environment, not an emergent property of the gene itself, activates expression of that gene. This is the crux, really. You know, if you said to me, what's one sentence that explains why age lock's even possible, it's right here. Because signals from the environment determine what genes are expressed and what genes are used. That's how you can produce a product to tell the youth gene clusters to be younger, because they're responding to the environmental stimulus. Okay? So genes don't turn themselves on and off. That's not their job. Their job is to hold the blueprint. The environment tells the genes which ones to use. They're not self-activating. They're selected in response to the environment. At a human level, that's perceptions, and this is a bigger talk too that we're not going to do tonight, but how a person perceives the world is a community of cells. So on a cellular level, that cell is perceiving its environment, but on a human level, when you're talking about 50 or 70 trillion cells, that community of cells perceives the world. A lot of it's through the nervous system and the skin, but those perceptions are determining what genes are being used to respond to the environment. And the reason this is really powerful is because it's not really what's happening, it's how you're perceiving it. That's why you want to work on yourself and your own perception of your environment, because it really is determining your biology. And that's why 5 people in the exact same situation are having 5 different experiences, responding differently. Some are having a powerful, loving, happy experience, some are scared. Very different genes are used in those 2 experiences. So blueprint. I put this here to show you, this is like your DNA, it's a house blueprint. It's not what is making the decision to build the house. It's the blueprint for the house. The environment's the determiner. Who reads this blueprint? How that person interprets the blueprint, what materials they decide to use. You see, I could have 5 entirely different construction teams build a house from that blueprint. They're not all going to look alike. They're going to choose a little different material. They're going to choose different size beams. You know, you just don't, it's going to vary from the same blueprint. And that's more like epigenetics. So Joe Chang gave me this slide to use. It's so potent. And it's displaying a lot of what I'm telling you. These are twins, okay? Twins are genetically identical. They have split. This kind of identical twins have split from one egg and sperm, come together and splits into 2. Exact same genetics. But they look very different. You know, one looks a lot older than the other. So right off the bat, you have to ask yourself, what's different? Because their genes are the same. It's not just genetics determining everything. And then when you look at their genetics, these are a chromosome from each of them, and it's pointing here because there's different gene expression happening there. You see, the basic blueprint is the same for them, but they're not using it the same way, so they look different. And that's because they've had different environments. They've had different food, they've had different sun exposure, different emotions, different thoughts, and they wind up looking different. And there's a cellular experiment to show a few of these examples too. Bruce Lipton is a cell biologist. He's been at Wisconsin and Stanford. And he took dystrophic cells, so human muscles that had muscular dystrophy, took cells out of there. He puts them in a petri dish in a healthy environment. Everything around there's healthy, for a healthy cell. And what was amazing was healthy muscle cells were made. And that's incredible because these cells were dystrophic. And they became healthy. And the only thing that changed was their environment. That tells you all along they had within them the potential to be healthy. And this is another reason we can do age lock, because the cell inherently wants to be young and healthy as long as it's in the right environment. It doesn't, you understand that? It doesn't make the decision. It doesn't have an opinion. It expresses the genes that the environment tells it to express. It's altering the biology of cell based on the environment or the perception, the beliefs. Another experiment he did, stem cells are like twins on a cellular level. They're genetically identical. He put them in 3 different petri dishes, and he changed ingredients in each dish. And what was incredible was that one petri dish turned into bone cells, one turned into muscle cells, and one turned into fat cells. Just by changing the environment. The genetics were identical. Okay? The blueprint was the same, but the environment was different. So then, they're working on now this epigenome project. Okay? The Human Genome Project was huge. And they sequenced all the DNA and they found 25,000 genes. The number of epigenome things is going to be massive. Because all those genes can be expressed so many ways, you know, and there's so many marks on the DNA to tell it to do that. I'll give you one more experiment. These mice are genetically identical. They both have a mutation that is, you're seeing it in this mouse. The only thing different between these 2 mice is this mouse, the mother was given extra B vitamins when this mouse was in utero. And he's completely shut off this mutation, this agouti mutation. These mice become overweight, obese, they turn yellow, they're more prone to cancers and diabetes. This mouse is perfectly healthy, brown, normal weight, but they're genetically identical. You see that? All that's different is that mouse got extra B vitamins in utero. And these guys were blown away by these results. It was this is the author, so Randy Yertle and Robert Waddell at Duke University did this in 2000. It was a little eerie and a little scary to see how something as subtle as a nutritional change in the pregnant mother could have such dramatic effects on the gene expression of the baby. Those 2 mice had the same genes, but they expressed them differently. The results showed how important epigenetic changes could be. Bottom line here, the point I'm making is you're not a victim of your DNA. It's all based on the environment you put that DNA in. You can change anything. Depends how you eat, depends what you put on your skin, what supplements you take, how you think. There's a great master once quoted as saying, the mind is the power. It truly is when you think about it. No pun intended. And you may not know this, but 90% of all cancers are not hereditary. There's no hereditary linkage. So yes, cancer can be genetically inherited. But that's not the whole story. Because people that are healthy for all their life, and then they turn 50 and they come up with some kind of cancer, and then their information is, well, it's genetic. Well, how were they healthy for 50 years then? That gene alone is not sufficient to give cancer. There had to be something else, an environmental change, because they were healthy all along. It's not just the gene. You see that? So yes, a person can be genetically predisposed, but there's more to the story. Often those people grow up thinking the same way as the parents did, or eating the same way, they're raised in that environment. There's a lot more to the picture than just the gene. So then we move into the age lock story. This is where it gets really exciting. Algorithm. I talked about an algorithm in the beginning. It's a finite number of steps to solve a problem. That's all an algorithm is. You've heard of it. Some of you have heard of algorithms from your math time. The example Joe Chang uses is Google, because it really makes the point. Google can search the internet really quickly and really accurately. Other search engines have other algorithms, but Google's is the best. And what does that do for Google? They dominate the market. Most people, when they want us, it's a verb now, Google it, But that's because they developed the best algorithm. Now, we have an age lock algorithm. We identify genes that change with age. target those genes with botanicals, with products, and reset them to their youthful expression. And I just showed you several of examples where the environment is determining how the genes are expressed. So if you find the right genes that change with age, and you target them appropriately with products that cause youthful expression, you can reset those youth genes. But that's a big statement, what I just said, when you really go to do it. There's 25,000 genes. This is the part where I'm going to say that's a lot, because it is a lot. You have to be able to study cellular gene expression. We have tremendous collaborations with very powerful partners. This is genetic code, okay? These are the letters of the genetic code. There's 3 billion of those. You have to know which ones are the aging genes. You see, this is a big job. So LifeGen Technologies is an incredible partner of ours now, exclusive partner, Dr. Weindruck, Dr. Prola. What did they bring to the party? Actually, over 30 years, they've been studying aging and genes that change with age over time. So the question I keep asking is, if you want to study genes that change with age over a 30-year period, how long does that take?
30 years.
30 years. And that's real time, because aging takes time. So LifeGen has brought to Nu Skin now a data bank of age-related genes everywhere in the body over decades. So even if someone wants to do what we're doing, how long is it going to take to collect that data bank?
30 years.
Yeah, and we're going to be 30 years farther on by then. I know these are difficult questions, but on top of that, they have a patent pending for the use of how they're doing their gene expression profiling. They were the first ones to use that microarray, DNA microarray, to look at the expression of the mammalian cells, which are mammals. They're going to patent their techniques. So on top of everything, someone else would have to find another way to do it. So here's the first thing. This first part of the algorithm, identify. It takes long-term research to do that, and we've named these youth gene clusters. We've named them because we found them.